For 40 years, most out-of-Africa theorists treated Asian fossils almost like an evolutionary embarrassment, pushing them into the background as isolated dead ends that supposedly contributed nothing to later humanity. At the same time, enormous pressure emerged to fold nearly every large-brained African Middle Pleistocene skull into early Homo sapiens, even when the anatomy clearly pointed toward far more archaic populations. Massive faces, heavy brow ridges, thick cranial vaults, and deeply divergent skull architecture were brushed aside in favor of simplified narratives. Yet the ancient DNA and protein evidence is now moving sharply in the opposite direction. Denisovan signals connect directly into Asian erectus populations, while fossils, once dismissed as archaic side branches, are drifting back toward the center of the human story. The old morphologists, the scholars who carefully traced anatomical patterns across Eurasia and Africa, increasingly look correct. The bones were preserving real population history long before genetics arrived to confirm it. Indeed, there is an uncomfortable possibility emerging from the newest ancient protein and DNA studies, and it strikes directly at one of the deepest assumptions in paleoanthropology. What if the old anatomists, the scholars who spent their entire lives staring at skulls, measuring brow ridges, tracing cranial arcs, comparing jaws, teeth, vault thickness, facial breadth and inner ear structure, were not wandering blindly through speculation at all? What if many of them were actually seeing something real long before genetics arrived to confirm it? Because something extraordinary keeps happening in human origins research. Again and again, ancient DNA and protein evidence are validating anatomical patterns that earlier generations identified using nothing but fossil morphology. Something strange is happening in the study of ancient humans, and the deeper scientists push into the genetic and protein evidence, the more an uncomfortable pattern keeps appearing. The old assumption that fossil anatomy was mostly subjective, mostly guesswork, mostly a kind of storytelling built from broken skulls and incomplete skeletons, no longer fits the evidence. Again and again, independent molecular evidence keeps circling back and validating patterns that physical anthropology had already hinted at through careful, mathematically driven morphology. What once looked speculative now looks disturbingly accurate. The deeper the ancient DNA revolution advances, the more it begins confirming that many ancient skulls really were telling the truth all along. This changes the entire atmosphere around the Denisovan mystery, because Denisovans were originally presented as almost the ultimate triumph of genetics over anatomy. A tiny finger bone from Denisova cave in Siberia produced DNA unlike anything known before, revealing an entirely hidden branch of humanity. There was no complete skull, no dramatic skeleton, no obvious face staring out from museum displays. Denisovans entered science as pure molecular ghosts, yet over time the exact opposite happened. Morphology began catching up. Certain skulls that older anatomists described as strange, transitional, unusually broad-faced or deeply archaic began drifting toward the Denisovan signal one after another. The molecular evidence did not erase morphology. It started validating it. And that is where this story becomes deeply unsettling, because the implications stretch far beyond Siberia. Researchers slowly realized that certain fossils already looked unusual in ways that aligned with the Denisovan genetic position. Massive jaws, broad faces, giant molars, archaic cranial architecture, and unexpected combinations of primitive and derived features suddenly became important again. Fossils that once floated awkwardly in the middle of the human tree no longer looked random. They started clustering around the Denisovan signal. This is where the newer studies become explosive, because the new enamel protein work from China does not simply identify isolated molecular fragments. It reconnects entire populations. Chinese erectus fossils from Zhoukoudian, Hexian and Sunjiadong now carry protein variants linked to Denisovans, pulling East Asian erectus directly into the Denisovan conversation. The old textbook image of Asian erectus as an isolated evolutionary dead end suddenly collapses. Instead, Eastern Eurasia begins looking like a continuous network of interacting populations stretching across hundreds of thousands of years. The fossils from China no longer sit outside the human story, they move directly toward its center, That matters because morphology had already been hinting at this outcome long before proteins entered the discussion. Fossils such as Dali, Harbin and Denisovan always looked difficult to classify cleanly. They carried large brains, broad faces, powerful brow ridges, thick cranial vaults, and strange mixtures of archaic and modern features. Researchers argued endlessly over whether they represented late erectus, archaic Homo sapiens, heidelbergensis, or something entirely separate. The uncertainty itself became the clue. These fossils never fit neatly because the evolutionary process itself was never neat. The molecular evidence now starts validating what morphology had been quietly suggesting all along. Eastern Eurasia contained deep, archaic populations connected to Denisovans. The remarkable part is that this pattern extends far beyond Asia. Once the Denisovan problem expanded into Europe and Africa, the old divisions between populations began dissolving. Cima de los Huesos in Spain changed everything. The fossils from Cima clearly show early Neanderthal anatomy. Their faces, teeth and cranial patterns place them directly inside the Neanderthal lineage. Yet their mitochondrial DNA did not align with later Neanderthals. Instead, it aligned more closely with Denisovans. Something does not quite fit there. If the Cima people looked like early Neanderthals, but carried Denisovan-like mitochondrial ancestry, then the separation between Denisovans and the so-called Heidelbergensis world becomes far blurrier than older models allowed. That single discovery quietly destabilized the clean branching diagrams shown for generations in museums and classrooms. The old image presented human evolution as a tidy sequence of forks, with one population cleanly splitting into another. Cima shattered that simplicity. Suddenly, a European population carried genetic echoes linked to Denisovans, while simultaneously possessing unmistakable Neanderthal anatomy. That means the Denisovan story did not begin in Siberia. It stretched deep into the Middle Pleistocene and deep into Western Eurasia. Then another strange pattern emerged. Certain skulls repeatedly identified through morphology as unusual, transitional, or difficult to classify kept drifting toward Denisovans in newer analyses. Kabwe in Zambia became one of the most provocative examples. Kabwe possesses an enormous face, huge brow ridges, a large brain, and a heavily built archaic skull, unlike later modern humans, yet also distinct from classic Neanderthals. For years, the fossil floated inside uncertain labels, such as Rhodesiensis or Heidelbergensis. Then, newer studies using gene regulatory phenotyping linked Kabwe strongly to Denisovans and to the Denisovan Neanderthal clade itself. Once again, anatomy moved toward molecular confirmation. Petrolona in Greece sits inside the same shadowy zone. Massive face, powerful brow ridges, archaic cranial structure combined with surprisingly advanced features. Bodo in Ethiopia carries a similarly broad and archaic morphology. None of these fossils fit comfortably into the older categories, yet together they form a recognizable pattern. When researchers began comparing predicted Denisovan anatomy against actual fossil morphology, many of these same skulls repeatedly surfaced as close matches. The implication becomes difficult to ignore. Denisovans were not simply an isolated Siberian population. They belonged to a gigantic Middle Pleistocene radiation extending from Europe into Africa and Eastern Asia. What makes this even more important is that Bayesian morphological methods repeatedly keep proving surprisingly accurate once molecular evidence arrives. Critics long dismissed morphology as too subjective, too dependent on researcher bias or artistic reconstruction. Yet Bayesian approaches transformed fossil analysis into something far more rigorous. Instead of relying on one dramatic feature, researchers compare large suites of anatomical traits statistically across populations. The outcome often predicts later molecular results with startling consistency. Neanderthal skulls provide one of the clearest examples. Long before DNA extraction became possible, physical anthropologists already recognized Neanderthals as a biologically coherent lineage through anatomy alone. Once nuclear DNA arrived, the genetic evidence validated that interpretation almost perfectly. The skulls had been telling the truth. Massive brow ridges, elongated cranial vaults, mid-facial projection, retromolar spaces, robust skeletons, and distinct dental patterns, all aligned with a genuine population history. That is the key point. Morphology fails when driven by ideology, assumptions, or typological thinking. It succeeds when handled statistically and comparatively across large anatomical datasets. The newer Denisovan studies matter because they emerge from precisely this newer generation of methods. Researchers are not simply eyeballing skulls anymore. They're combining Bayesian phylogenetics, geometric morphometrics, protein sequencing, enamel peptides, and genomic signals into unified frameworks. Independent methods keep converging on similar conclusions. This convergence now forces a re-evaluation of Eastern Eurasia itself. The old framework treated Europe and Africa as the engines of human evolution, while Asia functioned as an isolated side branch. Yet the protein evidence from Chinese erectus fossils undermines that picture completely. If Asian erectus populations contributed ancestry to Denisovans, then Eastern Eurasia preserved one of the deepest surviving archaic lineages on Earth. The fossils from Hexian and related sites no longer appear primitive in a dead-end sense. They appear ancestral in a continuing sense. That possibility transforms the meaning of Denisovans entirely. Denisovans stop looking like mysterious cave people hidden in Siberia and start looking like the surviving molecular shadow of an enormous Eastern archaic population system. Harbin, Dali, Penghu, Jiniushan, Hexian, and perhaps even some late Indonesian erectus fossils begin fitting into the same broader network. The line between erectus, Denisovan, and archaic Homo sapiens becomes increasingly unstable. The irony is striking. Ancient DNA originally seemed poised to destroy traditional fossil anthropology. Instead, it revived it. The skulls that many researchers dismissed as too incomplete or too ambiguous are returning to center stage because molecular evidence keeps validating their importance. The fossils were not random noise. They were preserving deep population structure across Eurasia long before genomes became available. This also explains why Bayesian phylogenetic trees have become so disruptive. Once anatomy is analyzed mathematically rather than typologically, surprising relationships emerge repeatedly. Asian erectus populations drift closer towards Denisovans. Denisovans emerge as sister lineages to Neanderthals and Heidelbergensis-like humans. Cima de los Huesos links Neanderthal anatomy with Denisovan-like mitochondrial ancestry. Cabwe and Petrolona drift toward Denisovan-related morphologies. Suddenly, the entire Middle Pleistocene begins looking less like a clean branching tree and more like a reticulating web of interacting populations. That web stretches across immense distances. Europe, Central Asia, Siberia, China, Southeast Asia, and Africa stop appearing as isolated theatres. Instead, they become parts of a connected, archaic world. Populations separated, merged, drifted apart, exchanged genes, adapted locally, then mixed again. The genetic evidence now shows this directly, yet the fossils had been hinting at it long before the molecules arrived. This is why the Denisovan mystery now sits at the center of the human story rather than at its edge. Denisovans are not merely another extinct branch beside Neanderthals. They expose the inadequacy of the old linear framework itself. Every new protein sequence, every new mitochondrial signal, every new Bayesian reconstruction pushes human evolution toward complexity rather than simplicity. And perhaps the most unsettling part is that the fossils still outnumber the genomes by an enormous margin. Ancient DNA survives only under rare conditions. Tropical regions destroy it rapidly. Vast stretches of Africa and Asia preserve almost no recoverable genetic material. That means morphology still carries much of the burden of reconstructing human history. If unbiased anatomical methods repeatedly prove correct once molecular evidence arrives, then many controversial fossils sitting in museum drawers today probably preserve real evolutionary relationships that genetics has simply not reached yet. That possibility hangs over every newly discovered skull from China, Indonesia, Central Asia, and Africa. The next major Denisovan relative probably already exists inside a fossil collection somewhere, classified under an outdated label because the molecular evidence has not caught up yet. The old categories begin weakening with every new discovery. Heidelbergensis fragments apart. Erectus stretches farther into later populations than expected. Denisovans expand across Eurasia. Neanderthals themselves look less isolated than once believed. Human evolution no longer resembles a ladder or even a simple branching tree. It resembles a continent-wide braided system of populations exchanging genes and anatomical traits over immense spans of time. The fossils hinted at that reality first. The proteins and DNA are simply arriving later to confirm what the bones had quietly preserved all along. Out of Africa, theorists spent decades trying to shove Asian fossils into the evolutionary closet, treating some of the most important fossils on Earth as meaningless dead ends because they did not fit a simple Africa-only storyline. At the same time, they tried forcing nearly every large-brained African skull into Homo sapiens, despite obvious archaic anatomy. Now the DNA and protein evidence is humiliating that narrative. Denisovan signals are pulling Asian erectus populations back into the center of human evolution, while the old anatomists who trusted skull morphology are being proven right again and again. The bones knew the truth before the genetics did. Because once ancient DNA entered the discussion, the neat categories began collapsing everywhere. Neanderthals mixed with Homo sapiens, Denisovans mixed with Neanderthals, Denisovans mixed with modern humans multiple times. Ancient Africans carried archaic introgression. Sema carried Denisovan-like mitochondrial ancestry alongside Neanderthal anatomy. The boundaries between populations became blurred almost beyond recognition. And suddenly, the anatomists who classified skull based on morphology, not modern political boundaries, no longer looked antiquated, and the stringent out-of-Africa total replacement theory looks like politically driven pseudoscience. In many ways, they looked astonishingly perceptive. Without genomes, without proteins, without molecular clocks, they still recognized that certain skulls formed meaningful biological groupings. They saw continuity between Eastern and Western Eurasian populations. They noticed recurring combinations of traits. They identified mosaic anatomy that later turned out to reflect admixture. They recognized that fossils such as Petrolona, Arago, Kabwe, Ciprano and Dali occupied important positions inside the human story rather than standing outside it. The irony is extraordinary. Ancient DNA was expected to destroy traditional anatomy. Instead, it revived it. The deeper science pushes into ancient proteins, fossil anatomy, and buried genomes, the stranger the human story becomes. Skulls, once dismissed as evolutionary dead ends, now stand at the center of one of the biggest scientific shifts in modern anthropology. Somewhere inside museum drawers, cave sediments, and forgotten fossil collections, more pieces of this hidden network still wait to emerge. And if the newest discoveries have taught us anything, it is that the ancient anatomists, staring silently at those skulls a century ago, were seeing far more than anyone realized. Thank you for watching. If these ancient skulls and forgotten populations fascinate you, stay curious, because the human story is becoming stranger with every discovery. The deeper scientists push into proteins, genomes, and fossil anatomy, the more the old boundaries between species begin dissolving. Somewhere beneath the mountains of Central Asia, beneath the caves of China, beneath the sediments of Africa and Europe, more pieces of this hidden network still wait to emerge. And when they do, something tells us they will not simplify the story of human origins. They will complicate it even further.